This is All Things Cancer, the podcast of the National Foundation for Cancer Research, hosted by NFCR's President and CEO, Dr. Sujuan Ba. Dr. Ba is a globally recognized cancer research advocate and visionary leader. All Things Cancer episodes will explore the hindsight, insights, and foresight of cancer, from research discoveries to patient care advances, and how NFCR and our collaborators are working to accelerate the process of making cures possible.
Welcome to All Things Cancer. I'm your host, Sujuan Ba, CEO of the National Foundation for Cancer Research. In today's episode, I bring you voices from the front lines of innovation in oncology. You'll hear from thought leaders focused on early detection and early intervention in cancer. Together, they explore the current state of science, the critical unmet needs facing patients and public health, and share insights on equitable access, emerging technologies, and the future of how we diagnose and treat cancer. Let's dive into these transformative ideas together. Our first speaker is Dr. Siddhartha Mukherjee. He's a physician-scientist at the Columbia Medical School and a Pulitzer Prize-winning author, one of the well-known voices in oncology.
Early detection comes in many forms. There's not one form of early detection. There is genetic testing. There is a mammography. It really depends on the kind of cancer. I also want to emphasize that early detection without a definitive therapeutic answer is not very useful. You can have lots of early detection, but if you can't do anything about it, it just causes anxiety and then you're stuck. So those two things need to move together. The success of trastuzumab was that it combined early detection with a very successful targeted precision therapy. So I think that it's very important that these things move together. That on one hand, we do early detection, that's very helpful, and that we also do use early detection in combination with a successful precision targeted therapy.
Let's now turn to Dr. Samir Hanash of MD Anderson Cancer Center. His team is redefining how we screen for cancers like lung, colon, and breast, shifting from age-based models to biomarker-driven precision. In a world where we test for cholesterol without thinking, he asks, why not do the same for cancer?
Most of our work is based on the application of proteomics. And in the past 20 years, the technology has improved considerably, not just down to the single cell level, but also the subcellular level. For example, we cover the cancer surfaceome exquisitely, identifying, not only quantifying proteins, but looking at post-translational modification. You have heard about neoantigens being oncogenes with a mutation. Neoantigens cover a much wider spectrum of changes in proteins represented by post-translational modification. So the work that we have done related to lung cancer, risk assessment, and early detection. And so as we all know, lung cancer is the number one cancer killer in the world. And at the present time, there are indications for screening for lung cancer for smokers who have smoked 20-pack years. But only 5% of those who are eligible are getting screened for lung cancer using low-dose CT. So what we've been working on for a number of years is to identify blood-based biomarkers that indicate a risk for lung cancer, whether you're a heavy-duty smoker, a light smoker, or even a never smoker. Can we find a marker panel in the blood that indicates your risk of lung cancer? And the answer is yes.
Could you explain why blood-based biomarkers are such a promising tool compared to current screening methods like low-dose CT scans?
I can't believe the work started actually back 15 years ago with mouse models of lung cancer. You would turn on the oncogene and then we would sample the blood asking at different stages of lung development, what can we find very early on? And so we identified a number of markers very early on during lung cancer tumor development. And the anchor marker for that was an immature form of surfactant B that we all have in our blood. But this is the prosurfactant B form. So prosurfactant B became anchor marker for developing a marker panel for lung cancer risk assessment and early detection. So we identified additional markers that ended up with a 4-marker panel to determine your risk of either harboring or developing lung cancer. And we have done extensive validation of this 4-marker panel in over 1 million subject cohorts, including cohorts in the United States and cohorts in Europe and elsewhere, showing that, in fact, it's very effective at determining your risk of lung cancer to trigger screening for lung cancer. Using the NCI PLCO cohort, prostate, lung, colon, ovarian cohort, showing that those that died of lung cancer in that cohort, had they gotten that test done early on and followed with a low-dose CT, it would have reduced mortality considerably. So we have not only evidence for risk assessment and early detection, but the gold standard for biomarkers, does it impact mortality? And we're able to show that indeed with a test like this, if applied to even a general population, you could reduce mortality due to lung cancer more than you could reduce mortality with current therapeutics for advanced stage lung cancer. So risk assessment, preventive intervention, and early detection are going to be very important in reducing mortality due to cancer. Not just finding the wonder drug for advanced treatment, which is like holding a tiger by the tail. Let's start at the very beginning with risk assessment. Who is at risk? We go to the doctor, we get our cholesterol checked. Well, why not go to the doctor and get your blood checked for cancer? We say, well, look, it looks like you have a profile that determines your risk for lung, colon, breast, and ovarian. And then that would lead to intervention.
That's an incredible vision for the future. For our listeners, how close are we really to seeing these kinds of cancer preventive vaccines in everyday medical practice?
I guarantee you, before the end of this decade, there's going to be cancer vaccines available so that if a woman is at risk for triple-negative breast cancer or breast cancer, here's the vaccine for you. Your risk profile should determine whether you should be screened for this cancer or that cancer. So your family history, your subject characteristics, and your biomarker panel or maybe genomic profile will be the major determinant for whether you should be screened for this cancer or that cancer and intervene to prevent the cancer in the first place.
Dr. Stephen Lipkin at the Weill Cornell Medicine is a board-certified clinical geneticist. He has been at the heart of identifying cancer predisposition genes. But what excites him now is the idea of intercepting cancer before it forms. The cancer dome, he describes, is a vision of the future, one where detection leads directly to action.
So certain individuals, certain families are of high risk for cancer, different cancers. So basically, this is what I tell my patients, there's about 120 cancer predisposition genes that we know about, of the 19,000 genes that cause a predisposition to a constellation of different cancers. It's non-random. So during my career, essentially I've been involved or privileged to be involved in identifying 7 cancer predisposition genes. That was very exciting, and it's wonderful to see patients who have a mutation in some of the genes that I was involved in identifying. But in particular now, pretty much, and I think Sapna next will agree with this, we pretty much know all, or almost all at least, of the cancer predisposition genes. There may be a couple that are lurking out there, but for the most part, it's done.
That's a really interesting point. For our listeners who may not be familiar, can you explain the difference between genetic and non-genetic cancer risk and why that matters for prevention?
So now, The question is sort of how do we best handle people with high predisposition to cancer? And then, of course, there are also non-genetic, predisposition reasons. So, for example, we're talking about lung cancer. Lung cancer pre-malignancy is a very clear and sort of established, group. Individuals come in, they, for instance, they have a car accident, they had a CT scan, they have nodules that are found, or non-solid, what are sometimes called non-solid nodules. It's unclear really sort of how you best handle these, but they do progress to adenocarcinoma. So what do we do about it? So the focus that we tried to take, and we have an NCI Center for RNA cancer vaccines or immunoprevention vaccines, or actually it's immunoprevention and interception vaccine. So the concept is really to try to use cancer interception and a concept that we call cancer dome. The cancer dome concept essentially is you try to detect as early as possible. And once you detect, you try to intercept with vaccines. So the idea is to try to use cell-free DNA to the modalities, identify some of the aberrant proteins, and then try to develop their ways now, working with Drew Weissman, others who won the Nobel Prize for COVID vaccines, ways to sort of to tweak the composition of RNA vaccines, and the lipids and the lipid carriers and the RNA itself, actually. And you can do vaccine co-cargo. There are other things you can put in, for example, various cytokines. There was a recent FDA approval for N803, which is sort of a modified form of interleukin 15 that's used in non-muscle invasive bladder cancer. So there are ways that you can actually, detect if you find some of these things on cell-free DNA. You can not just say, oh, this is bad, but and angst out your patients, but that you have the potential actually to make these more immunogenic and recognized by the immune system. So once again, the idea is intercept or detect, intercept, eliminate.
From gene mapping into interception vaccines, Dr. Lipkin's work proves the future of prevention isn't hypothetical. It's already in motion. We asked Dr. Sapna Syngal to share her views on early detection and prevention. Dr. Sapna Syngal is a professor of medicine at Harvard Medical School, director of strategic planning for prevention and early cancer detection at the Dana-Farber Cancer Institute. She brings an incredible analogy. We don't wait for a heart attack to treat high blood pressure. So why do we wait for cancer to develop before acting? Her work at Dana-Farber is a blueprint for how to implement precision risk tools at scale.
An analogy that I use all the time is we would never wait for somebody to have a heart attack to check their blood pressure and their cholesterol. And yet that's what we do for cancer. I think we wait. Our approach to cancer is very reactive rather than being proactive. And this has been a fundamental issue in cancer care, for the longest time. And I think sort of gladly, I think we've been changing, largely, I think, first precipitated by our discoveries in hereditary cancer. So I do think that we still have a lot more to do to implement what we already know. One of the big challenges of the genomics, precision genomics, is how are we going to get clinicians to actually implement this in general practice? So we've developed these risk assessment models for inherited predisposition called the PREM models. And we just got funded to actually put PREM within the EMR of a Kaiser group in the Kaiser Mid-Atlantic, actually here close by to here, Denver Health and Logan Health. And they all serve very underserved populations. And the idea is that everybody will get that tool sent to them before their doctor's appointment. They'll fill out the survey, they'll get their risk prediction, and they will have the option to schedule their genetic testing before they even see the doctor, and then get called with their genetic test results. So I think that's the kind of wave of the future that we need to have. And there's no reason why we shouldn't be screening people for their smoking history and then setting them up for their CT scan, right? One of the biggest problems we have right now, we know CT scanning works for lung cancer screening, but only a minority of the population is actually getting it. So implementing the tools, I think, of what we already know is I think I think we can surely do that in the next few years.
When risk tools are embedded in electronic medical records and paired with outreach, they become powerful equalizers in cancer care. Now we hear from Dr. Ajay Goel, professor at the City of Hope. His work on gastrointestinal cancers highlights how science is getting closer to detecting cancer earlier than ever before, and not just in its early stages, but in its pre-cancerous phases. He also reminds us that if detection tools aren't affordable or inclusive, we are just reinforcing existing disparities.
So my research is mostly focused on early detection of gastrointestinal cancers. So within that, I was early on trained with a gastroenterologist at Poland. So I started my career on studying colon cancer, but over the years, I began to study all GI cancers, including colon, pancreas, stomach, and others. And a big part of my research for the last 2 decades have been on developing biomarkers, cell-free DNAs, RNA, other biomarkers for early detection of cancers. In the last decade or so, we have spent quite a bit of time on studying extracellular vesicles and tumor-derived exosomes, because we have learned that tumor-derived exosomes may have analytes which we can use for earlier detection of these cancers. In addition, we also studied the basic biology of RNA, DNA, and also trying to understand the role of gut microbiome. So we all know microbiome is very, very important in terms of cancer pathogenesis. So we try to look at the interplay between gut microbiome and cancer etiology, as well as possibly using gut microbiome also as a possible biomarker for early detection of these cancers. And a very small interest given the disparity of these cancers, especially in a population like the US, where we are different ethnic and racial groups. So we also study racial disparity and biomarkers for finding these cancers in different racial and ethnic groups. Yeah, so I think when it comes to early detection, I think we have come a long way in terms of detecting many of these cancers. But I think when we look at individual cancer, we still, just like you said, we haven't done a good job. Colorectal cancer, very common cancer, we know that it is preventable if you can find it early, right? So even now, even the data from this year, 50% of the population in the US does not get screened by colonoscopy, who are screening eligible, but they don't get screens. So I think there are multiple reasons. One, access to healthcare, people's personal beliefs that they need or don't need, and then the cost. And I think in terms of colon cancer, the best test we see, or one of the best tests we see on TV screens, Cologuard. So I think that's a wonderful test, but still, I think the compliance and participation is still not as high as you would like to see. So if you look at the data even now, I think most of the early detection tests, although we brand them and say these are early detection tests, but I think if you look at the data carefully, most of these tests perform well still in the advanced stages, not in the early stages. So if you look at the Cologuard data, even from 3 months ago, published in New England Journal, so this test still has, very poor sensitivity for advanced polyps, advanced adenomas, or stage 1 cancers. And I believe those are the true lesions we should be focused when we talk about early detection, not a patient who already has a stage 2, 3, or 4 cancer. So we have done quite a bit of work on this. We are actually working very, very closely with Cologuard. So we have a test based on cell-free DNA and RNAs, which has almost 90% sensitivity and specificity for finding patients with advanced polyps. So I think those things are going to change. Similarly, we have a test for early detection of pancreas cancer, which we are almost about to be published now. So I think the focus for early detection, we have to really mean the word early, means earlier and earlier precancerous lesions or stage 1 disease, not later stages. And I think once we can do it, if the test is affordable, it's a blood test, I think we can make a bigger difference compared to looking at some of these tests which really don't work in the earlier stages.
We say early detection saves lives, but Dr. Goel urges us to ask further. Early for whom? The answers must include every community, not just the ones closest to the major clinical centers and hospitals. I invited Dr. Carlos Doti, Head of Medical Affairs of the U.S. Oncology Business Unit at AstraZeneca, a hematologist by training, to offer his views for us on early detection and early intervention for cancer. He offers a rare but refreshing industry view.
We started the journey at AZ in 2012 when we changed our strategy to be more focused on oncology. And we started our journey by entering into the metastatic space in lung cancer and in ovarian cancer. And it was clear to us that the only way to be successful as part of the stakeholders in cancer care was moving into earlier lines. So we pulled all our assets into earlier lines, and you see consistently in every area that we work, we have trials going earlier and earlier and earlier. In lung cancer, we move from metastatic to the unresectable, and then we go to adjuvant, perioperative, sometimes more effective than others. But of course, when you see that from a continuum of a company, not investing in early detection and prevention is leaving your work in half. And we even asked internally to our CEO, so what if we're successful and we actually eliminate cancer? And I said, so be it. This is our objective as a company, is to transform care. And transform care is not just bring better treatment to a subpopulation, but actually to all patients, right? And we know that not every risk assessment or every early intervention will be 100% perfect. So we still work in every single area to transform care. This is very personal, but I'm a hematologist, but I'm a cancer caregiver. I'm a caregiver to a cancer patient. It's my mother, and I lost my father to cancer. So how can I not think about preventing what could have changed my family and for every other family out there? And I think this is the driver, is to have an open conversation on, pharmas are not non-profit organizations, but how we have an integrated conversation on how to transform cancer care from the very beginning. And that's us.
When industry prioritizes transformation over profit cycles, we all win. Dr. Doti shows what it looks like when industry ambition aligns with a human need. To wrap up, I ask our panelists to respond to some of the most pressing and thought-provoking questions we hear across the field, revealing the huge challenges we all need to confront in order to improve the current healthcare system. The first question is, why aren't we using more personalized risk profiles for screening?
How do we screen for lung cancer at the present time? It's based on smoking history. 20-pack years represents roughly a 1% risk of lung cancer. So the challenge is, for other cancers, can we come up with a profile that represents a 1% risk for that cancer? A 1% risk for pancreatic cancer then would justify wanting to screen for pancreatic cancer. It's not a rare disease in that 1% that are at risk. It's a rare disease in the general population. And so the challenge is, through a risk profile and a biomarker profile, come up with a 1% risk for breast cancer. And for that woman, you better get mammography, or even better yet, get MRI. And if your risk is low, you don't need to get mammography every year. And there's a lot of concern right now in other countries about, mammography is not cost effective. And so, but if we can identify personalized risk, that's the criteria. Personalized risk for this cancer or that cancer, it will make it much more beneficial, cost effective, and reduce mortality.
Even when we have tools that work, why is uptake still so low in underserved populations? And how do we fix that?
So I think it's a great point. I think we all touched upon this in our comments, is that there's lots of knowledge that's not being implemented. And that's one of the biggest things that we do have to fix. That doesn't mean that we don't do other things simultaneously. So implementation of average risk screening. We can certainly do a better job, but there are a lot of efforts in this space in terms of getting out there in the communities, getting out there with the education and making people I think we have to approach it in the same way that we did with COVID. Remember, at the beginning, nobody was getting vaccinated, underserved populations. When you actually started to reach out to the populations by people from their own communities, the vaccination rates in Blacks and Latinx individuals went up. And in the same way, we need to do that, in our general population. From the risk assessment part, it's even more complicated. And we heard this. We wanted to increase access in our diverse populations. We had very low uptake from Blacks and Latinx populations. And they said, we're not going to do this because in my state, I can't get the surveillance covered, right? My insurance doesn't, Medicaid doesn't cover surveillance. So I think this has to be a multi-pronged approach at not only getting the risk assessment done, but also making sure that people have access to the interventions that they need downstream.
Do we need to rethink how we engage people with tech tool, education, or incentives?
I think reaching out to patients once again and thinking sort of like, what do they want? So being able to, once again, use some of these machine learning tools to be able to communicate with people and bring them into the system. We all know, obviously, that these tools have the AI tools sometimes to say things that aren't true. So there's an error rate, and but this can be improved. So reaching to people, where they want to, and try to kind of encourage them at least to come in. And then obviously, comparing colonoscopy and liquid biopsy, as we've heard, it kind of thinks is another way. And is that going to solve the problem? No, but can it make a dent? I think so.
Whether it's increasing uptake, improving reimbursement, or ensuring easy access, this work doesn't stop at discovery. It continues in the system we build around it. We close this episode with one final note from Dr. Mukherjee. What he shares here is profound. Early detection alone is not enough. It must be paired with effective, accessible treatment. And when he talks about slashing drug costs using AI and distributing life-saving medicines globally, he is challenging not just science, but the system.
The CDK4/6 inhibitors cost $24,000 to $30,000 a year. That's just their cost. You can triple that, quadruple that if you add in the medical therapies, the oncologist visits, all the various things that come to it. I will challenge you, and I will challenge myself, that in 5 to 10 years, I will produce that drug using AI for 1/10 to 1/20th that cost. And I will distribute that drug. It will be protected by IP, so not challenged by a multinational company. And I will distribute that drug across the world so that women in North Africa, South Africa, women in Brazil, women across the world have access to a version of that medicine across the world. And why do I know I can do this? Because I am making trastuzumab at 1/20th the cost. So I will challenge myself. I will come back to you in 5 years and I will tell you I have made a CDK4/6 inhibitor at 1/10 to 1/20th the cost.
You have just heard from some of the most forward-thinking minds in cancer research. If there is one message, it's clear here. Early detection is not only a technology issue. It's a coordination issue, a compassion issue, and a willpower issue. At NHCR, we are bringing together the scientists, the clinicians, the strategists, the business leaders from the whole ecosystem of cancer care that will shape the future of cancer prevention. That includes supporting promising advances like a potential breast cancer vaccine, as highlighted by Dr. Hanash, and championing tools that are already saving lives, like a Cologuard, which Dr. Goel reminded us is helping more people detect colorectal cancer early from the comfort of home. We encourage everyone to talk to your doctors about scheduling regular screenings, including mammograms, pap smear test, and the colon cancer test. Many of these early detection measures are fully covered by insurance policies. Until next time, stay informed, stay inspired, and stay with us on this journey.
Thank you for joining us for this month's edition of All Things Cancer. Please subscribe so you don't miss future episodes. Learn more about how NFCR is working every day with the cancer research community to make cures possible at nfcr.org. You can also reach us on our call line at 800-321-CURE.